Testing 1 2 testing. Testing 1 2 testing. Testing 1 2 testing. Okay, well, I think everyone can hear me okay. Did a couple of quick audio checks. Glad to be back on the great hike yesterday, looking for some more mushrooms and other fungi. Hey, Manny, how's it going? And had a little break in the rain, so went for a hike, slipperiest damn trail ever. Hiking boots would not have even helped. As you took a step, the whole ground just smushed below you and you slid out because you're at an angle. So I bought the equivalent on Amazon, these basically equivalent of mud crampons you attach to your shoes. So the next time I go hiking looking for all this stuff, I won't almost fall on my face 20 or 30 times just walking down the trail. Super, super slippery. Anyways, trying to get to the summit before it got dark and come back down and get some samples. And saw one of these guys in the bushes, basically. I have another shot of it here. Little rough skin newt. There's a California newt is sometimes more common. It's not as rough looking. They both have the bright orange warning coloration on the bottom, which is very diagnostic of the poisonous salamanders, just to warn predators that don't eat me. You're gonna waste time eating me, spit me out because I taste bad, and if you do eat me, you're gonna get sick or possibly die anyway. So warning coloration works quite well. It's really cool about these guys, as you can see, the eyes have that kind of a almost day glow green yellow color. Very striking. They're just cute little guys walking through the wet debris in the forest. This is later in the day, so they come out at night actually and do this kind of mass gathering, basically out looking for mates, essentially, when you see so many of them out like that. I only saw a couple that night. I think I have another picture of it here. The sun had already started to go down, but there's still plenty of light. And I think, let me verify I got the right image folder here. This is a shot of of course, at maximum zoom. That's not fun. There's one on the ground going across the trail. When it saw me, it goes, oh, there's a big shape coming my way. I'm heading the other way. And you can see the incidental light, the grazing light, because the sun is actually going down. Hitting it from the side. So that part of the trail is not very wet, but it's heading over to wet areas. These, salamanders are basically amphibians. So, as you know, amphibios means double life, because they start as a water dependent creature living in the water with gills, and they in the case of frogs, they start as tadpoles and then legs bud out, and then they breathe water, then they eventually start to breathe air, and then they come out, but they still have to live near water because the skin has to stay moist, and their offspring are generated and laid eggs in water, so they need to be near water at all times. Some toads are kind of extreme. They can go into dry areas, but salamanders, same thing. In fact, some actually breathe so much of their skin they don't really respire much through any kind of lung apparatus. Haven't read about them for a while, but they're so-called lungless salamanders. You can actually find them at the bottom of your pool. Sometimes if you have a swimming pool and there's not too much chlorine, they will actually be down there and not be dead. You have to fish them out of your pool. So this guy was heading back the other direction, and if I had stayed later when it was dark, there would have been a bunch more going on in this mass walk. I've been on a trail at night before where they had so many that on a hike, it was a night hike, to look for owls and other nocturnal raptors. And the light started hitting the trail because it's muddy and we want to see where we're walking. And I saw this motion, and there's tons of these guys going across the trail in mass numbers. And unfortunately, a bunch have been stepped on already because no one was looking down. They're looking up, looking for birds. And so yeah, someone got stepped on, which is really sad. But there's so many of them. The ground was like alive with them going across the trail at night. You ever saw the animated film Allegro non troppo by Bruno Bozzetto? There's a great scene showing this silly kind of version of evolution as animals walk through this long period to Ravel's Bolero, which is really cool. And just the way they're all kind of marching to the beat in mass numbers, it was kind of like these guys going across the trail. It's kind of fun. And I can just show you, let's see if I can share this real quick without freaking out OBS. There is a, I don't get to the microscope. There's a, let's see, I'll change it to this app here. There's a cool little video of this guy I got walking just before I picked him up to get a nice picture of him. And I hope I can get this to connect without freaking out. Let's see, you are connected to, no, not that program. Let's go to the other program. Properties. Let's see. And then we'll drop this little movie file in here and you can just see them walking. Get this out of the way. Here's a short clip of the guy marching around. See if that shows up okay on the stream. I might have to play it once or twice if it didn't launch right away. Did it get cut off? Or did it even show? I'm seeing a black screen. That's not cool. OBS isn't being cooperative. Let me try this. I'm going to... Well, tell you what, I'll link a separate video on MicroAddict after the stream, and you can just see it's just a short clip of the guy walking through the debris, and it's all moist, and he's just doing his little marching movement, which is kind of stiff. I'm not sure why OBS is being a pain in the butt, but anyway, I'll go on to the microscope stuff. That's what we're here for. Discard that. Turn that on. Okay. Yeah, sorry man, I got a late start. It's just I had a bunch of stuff going on, and I had these samples I collected yesterday. I was going to do a stream last night, but I had to make that hardware jam thing, and I didn't even barely had time for that. My Italo disco thingy. So I wanted to use these samples while they're still fresh and things are still alive in them before they lose all their oxygen. So let's go in for a close-up here. And I'll grab your audio, Manny, for a future stream, since you said it's okay. I'll make a nice ambient Dr. Synth stream for the wild critters. Let's do fix this up here. Look for some goodies. I got a white balance issue. That's always fun. Why is that doing that? That's the wrong color. Okay. These are just 3 different water samples from the place where I found the Newt. One is a massive puddle that's been persistent for a while. One is from the actual creek going through a local place called Rancho San Antonio hiking spot. And one was basically a pipe, or a runoff pipe near that drains into the creek. Just wanted to see what was possibly coming out of that. And this is from the puddle. Very, very large puddle from all the rain. And so far, not very interesting, other than some shapes. Not seeing a lot of individual movements. It's more like a star field than anything else. Whole lot of debris. But you never know. This is the kind of area where you could find possibly an amoeba or a paramecium. All those little yummy debris bits to chomp on. Lots of colorful stuff here. A whole lot of colorful stuff. Nice shiny little air bubble there. Not seeing any movement, just debris. Gosh, tons and tons of debris. Oh, here we go. Nice. Yay, looks like another little Stylonkia thingy-majig. It's got the little spikes on the ends. Fix that. Oh, no, I'm wrong. I thought I saw spikes. It's actually just a diatom. God, that white balance is way off on this thing. What's going on? Holy moly. Why is it doing that today? And my white balance gets so wrong. The diatom's moving though. This is a single cell plant on the move. And it's got a very firm casing around it, made of silica. You can actually make out the little ridges in it. It has kind of like a toothed edge on it, like a comb. I don't know why this image is kind of fuzzy. Let's see if I can fix that. And this kind of, well, it depends what angle when they rotate. Some points of view they're bilaterally symmetrical, some are radial symmetrical. Some are very, very odd. I have only one bit of footage I ever got of one. If you've ever used one of those old-fashioned tape measures that are wood, that fold up and then extend out, and it's kind of flexible, so it doesn't. If you hold it straight out, it kind of bends under its own weight. But the concept is this series of segments that folds into itself. You can put it in your pocket or into a backpack or whatever. Kind of old-fashioned tape measure before they had the actual, nice metal or plastic rolled tape measure. Anyway, there's a diatom that does that, but instead of these segments folding back on themselves, it actually slides and pivots on those segments. So you can get it extending way off screen, like sliding glass door bits going past each other, but only going so far because they're still connected. And it's very interesting, weird, massive colonial diatom thingy. I'll have to post that footage one of these days. I haven't had a chance to edit it. Actually, I almost lost it. The stupid app crashed when I was recording it, and I think I was recording it on my phone before I got this other camera that's USB, and so I had to use a recovery software to actually rebuild the video file. Otherwise I would have lost it forever. But it's a trip seeing the segments bend and flex and turning, and some of it's poking out at you in the Z direction. That's almost impossible to focus through the whole thing, and some of it is extending out. Oh, there's another buddy at the bottom. More diatom spaceships coming in here. And you can see the tint They've got some kind of a yellow-greenish color. The typical diatom chlorophyll is more on the yellowish looking color than the green. And they also tend to have these oil droplets in them. You can see those two big blobs in there. Those are usually these kind of oil droplets for storage, since they can't basically store food all the time, and they're not that big, and they're not always getting light for photosynthesis. They have to have some kind of a food storage mechanism. So, and it could be some other thing too. I'd have to read up on it. I forgot. The diatoms are so bizarre and wild. I forgot all the uses of the oil droplets. I think it's mostly for food storage. They're going to crash. Bam! But you can see how they're firm. When they hit, they don't flex and bend around each other like a paramecium does when it hits another paramecium. They're just hard shells. And that is why you get... they've been used... they're not used in toothpaste. That's calcium carbonate. But the silica stuff is used in all kinds of like industrial stuff, even as a filtering agent. It's essentially glass powder, so you wouldn't really want that in your gums or whatever. But diatomaceous earth, it's called, and it's used as an element in your pool filter. That white powdery sludge that you have to change out periodically on a pool filter. It's millions and millions of these guys, skeletons essentially. Hey Lone, how's it going? Got some diatoms here. This is a giant pond. I was collecting stuff. If you missed the beginning of the stream, I had a shot of a rough-skinned newt from a hike I did yesterday up in the hills here. It was kind of off and on pouring rain, muddy as hell trail, big mess. Almost fell on my ass about 20 times. A couple times I really slipped and really caught myself. Otherwise, I could have gone flying off the edge of the trail into a bunch of poison oak and stuff, which would have been wonderful. Anyway, so the yeah, so I was saying about the diatomaceous earth is it's millions and millions of these little guys making that white powder, and even more amazing is that the White Cliffs of Dover are giant calcium carbonate deposits, prehistoric calcium carbonate deposits from these calcium carbonate shelled creatures called foraminiferans, which I haven't had a specimen of those in a long time. They tend to be more saline, so like brackish water or salt water, like at the salt marshes or the ocean. I'll have to start getting some more saltwater samples soon, because there's some good stuff in there. You can find radiolarians and foraminiferans and all kinds of dinoflagellates. Dinoflagellates are a lot of fun. But they're also nasty. They're the ones that cause the red tide, which is why the filter feeders like clams and mussels and stuff eat them, and they become toxic. You can't eat them because not only will you get sick, but it could possibly kill you. The toxin buildup of the dinoflagellates and their toxic compounds in the body of the mollusk will really screw you up. But I haven't eaten seafood for a hell of a long time, so I'm safe. But people who are constantly doing that, or especially during when it's a warning for red tide, it's like Russian roulette. It's very scary. So nice, got some little diatom action. I haven't seen one of these little boxy ones for a while. Usually get the ones that look like cigars, these tapered ones that are rounded on the edges, or you get various other kind of blobby rounded ones. There's some beautiful artwork if you go online and look for diatom shells, or they actually call it a test sometimes. Beautiful artwork done by, microbiologists or just, bio art people taking pictures of things, of just the shells, not the live animals. And in various lighting conditions. Some are done with photomicrographs from an electron microscope. And just the structure and the beauty and the intricacy of these things, it's just crazy. And they're just, single cell plants moving around. Hey Robert, how's it going? Last minute late stream, but it's good for the folks that are not in the US at this point. It's just nice to catch you guys once in a while. I know Wagu's always up, but either staying up all night or just getting up around this time. So this, I wasn't sure about this sample. Got some good diatoms here. You see these little ridges. There's some of the ones that look kind of feathery, where there's a central spine with some kind of ridges. It's actually called the rafe. And there was apparently debates for a hell of a long time about how these guys did their locomotion. They weren't sure if it was ion propulsion of some kind, pumping ions and basically causing a electric potential gradient to make them move to and fro. But they weren't quite sure. But then they started studying the shell a lot more and the structure, and you see these like ridges in the rafe, and then on some of these have the kind of toothed edges on them. And oh, that's interesting. Is he smushing something else or something gelatinous here? What is going on there? That is not just a well, maybe it is a diatom making all that movement. Sometimes it's hard to tell when it's gelatinous. Anyway, so the last thing I read several years ago is they're doing some kind of basically the equivalent of like a snail's slime trail. There's like a microscopic ooze that comes out. This is kind of mucilage stuff that they exude from the rafe or from the other ridges in the shell, and they're gliding on a thin covering of this mucilaginous stuff. Or the action of exuding it is pushing them along. So it's complicated stuff. Yeah, this guy's pushing through his debris. Because it's a hard shell, they can just force their way through, just like a submarine going through a bunch of muck. Yeah, we get newts, salamanders, all kinds of stuff here. I was just saying earlier, we used to get them in our pool all the time. The ones I call lungless salamanders, because they breathe primarily through their skin. The skin is constantly moist, and they're pretty low-key, not very active animals, so they don't need a complex respiratory system. So they can actually breathe, just like your skin breathes all the time. Like if you put full body makeup on you and cover up all your pores, you can actually get really ill because your skin does need to respire, even though you primarily breathe through your mouth and your nose to your lungs. Your skin needs to breathe to a certain extent, and in the case of the salamanders, they can pretty much have once they're adults and they breathe air, they can't breathe in the water like they did when they had the little gills sticking out. Like you've seen those axolotls before. Those are the ultimate fun little salamander relatives, because they're actually has to do with the thyroxine and thyroid compounds and stuff, and when they decide to actually lose their gills and become adults in quotes, because they actually are adults, but they are retaining juvenile traits. So you can have a big axolotl salamander in the water, sitting there breathing with these big bushy gills, but it's technically an adult. And because of the concentration of thyroxine and stuff, so it's a thing called neoteny, where they actually retain juvenile traits in an adult form. It's very interesting stuff. So yeah, love these guys. But yeah, I used to have to fish them out of the pool all the time, the little lungless salamanders. And we didn't chlorinate the pool that much because I used it so much, and the rest of the family did as well. So I didn't want it to be like the pool at school where your eyes are constantly burning because you just used too many damn chemicals. So the animals are just fine in our pool, which they kept going in there. So, all right, that's good for the. I mean, I'll go for a quick. Nah, I'm not gonna. It's moving too much for the super close up. This is 400 power right now, but I'm gonna skip the 1000x because I want to find some more goodies. I also have some physical specimens to look at on the dissecting scope. So let's see. Let's grab another sample. This is from the creek nearby. This is the creek. Just make sure this is. There's a little trick you can do sometimes to maximize your samples. I actually built a rig with a little cordless screwdriver or drill to slowly or fairly quickly spin your specimen tubes, kind of like a centrifuge, but poor man's centrifuge, and it concentrates most of your goodies toward the one side of the pointy collection tube. But I don't have it all set up right now, so I'm doing the poor man's version, spinning the tube on the table. So let's try this sample. What do we got here? The water here looked pretty, like there was a lot more debris obviously in the water I just showed. This water looked, because it was running water from a creek, it was pretty clean and not very busy, so it might be a fairly boring sample. We'll see. You never know. Got the damn white balance issue again. Stupid thing. Alright. Let's see what we can find. Also got some more moss. And I'm going to soak shortly for some tardigrade action, hopefully. Yeah, one of my favorite salamanders in the area is Pacific giant salamander. So it has a great genus, Dicamptodon, and they actually bark. It was kind of noise when they're in the, and they're also pretty damn aggressive too. So you can pick them up, kind of like a tiger salamander in the pet trade. We had tiger salamanders at my zoo I used to work at. They are pretty cute, and you can hand feed them if you're careful, but if they miss and they clamp down on your finger, they're very strong, and they don't just let go right away like, oops, that wasn't right. And you can actually, just like how a frog can bite you sometimes. There's some funny videos online of guys playing with these little apps on their phone, which make the have these little bug squash games where you tap into bug squashes. And the frog sees activity. This guy's got his little bullfrog, basically, trying to play the game by hitting the stuff with his tongue on the screen. And he's laughing, and the frog is doing its little business, not getting anything because it's just a stupid screen. And then he tries to do something on the screen with his finger, and the frog goes for his finger and clamps down on it. That's pretty funny. So lots of little cell bits and stuff, but nothing Too interesting here. Again, this is pretty damn clean water. It's running water, so I didn't expect to find a lot. But I did stir up the kind of little bit of dirt at the bottom of the rocks just to get a little extra stuff, some benthic stuff. Interesting cluster of stuff there. Yeah, what's going on there? Yeah. Got some osmotic gradient going on there. Make it look like stuff's actually moving, but it's so this water going past the I put too much water on the slide, so it's going past the cover slip. So it's just water shifting around. There's no real proper movement of life. Just keep searching. Some little plant bits there. Plant rootlets and such. So far, a pretty boring slide. Lots of various cell bits, parts of plants and such. No movement. Some colorful bits. Oh, what was that? Oh, nothing. Some of these things look just like little amoeba structures, but they're just gelatinous groups of cells. What is that on the end there? It's right out of my cell room, my scan range on this thing. Yeah, it's a pretty boring slide. So a quick one through just to see if there's anything. It's not just debris. Alright then, that was a dud. Can't always have success. It's part of the deal. Let's try another one here. Let's go to sample 3, which is the pipe runoff. See what we can get here. Yeah, the Pacific giant salamanders that we, for some of my classes I used to take, we'll go out and do field study, and there's this beautiful place at Point Reyes. This place called the Point Reyes Bird Observatory. It's an official place where they do bird banding, bird analysis, bird research, all kinds of thingies. And there's a little trail that goes down to this giant crevasse. It goes all the way down to the floor of the crevasse, right by the ocean there. But it's separated, so it's just this little kind of freshwater haven, just moist. We nicknamed it Fern Gully. It looked just like something out of the movie Fern Gully. And you go down there, just ferns all up and down the walls of the area, and there's a little creek running through it, fresh water, and you can find the Pacific giant salamanders in there. It's pretty fun. Alright, what do we got here? This is the runoff slide. There's the end of my slide. I'm on the wrong area. There we go. Where'd my cover slip go? It's weird. Cover slip shifted. I got another one on there. Okay. Let's see what's going on here. Ooh, that's way overexposed on this guy. Take this down a bit too. A little too much exposure. That's still too much. It shouldn't be so bright. That's a little better. Okay. Now it's all blown out. More plant debris. Let's kill this white balance. I got that little stupid thing on the screen there. Yeah. These samples aren't too crazy. Again, running water and pretty clean looking water. Not a lot of stuff going on when I collected it. Oh, there's some algae there. Look at that. That's nice. Might be a little piece of moss that broke off. Let's see. Cries out for more magnification. Let's see the cell structure there. Not seeing any cytoplasmic streaming. The cells are pretty boring looking, but there are globules in there. Kind of make it out a little bit. Maybe I can zoom on this a little bit. There's a digital zoom here that helps a tad. You can see the globules inside these cells. Basically chloroplast and other bits. But no action. Let's go back a step here. bits next to it. Get a nice view of the cells there. I think I have to clean this optic. It's still kind of fuzzy. Still a bit hazy. Maybe you can see the cells all lined up there. What else? What else? What else is going on here? Some crystal growth there, little jaggedy bits. See water forming the water got on the outside of the cover slip, so you get these little really cool, showing the little islands of water and the bumps and the surface tension and the cohesion of water that shows. It's very, very thin at the as it tapers off, but it has a bulge. Lots of little bulges here, little islands. It's pretty cool. All the properties of water. Yeah, I'm not seeing any cool life to look at here. Just junk. Lots of interesting junk bits to look at. So as an example of water that's either hasn't been sitting long enough to collect a bunch of life, or was just not interesting enough to sustain any life, not enough nutrients in it or whatever. So that was the water samples. Let me switch to the dissecting scope for a bit and look at some big things. So first let's look at the moss, because I want to see if I can find any. Just look at the nice quick look at the moss before I soak it to get the tardigrades activated. It's an interesting structure to look at just as is. It's quite scaly. Kind of like a Norfolk Island pine. Micro Norfolk Island pine, where the leaves kind of sheath on a central axis. The margins where it's a little easier to see. I can also do a little backlighting, which sometimes helps. Not so much in this case, a little bit. But you can see boy, moss easily rehydrate so well, because all you have to do is basically put a drop on it. This is not that dry, it's still a bit moist because I kept it sealed up so it wouldn't air dry. But what's fun about it is if you take, for example, I get the tiny specimen with this big dropper in here. If you watch the water, drop on it, of course it's reversed on the microscope. There's a drop of water. See how the water courses through the surface tension of the water and the capillary action of the leaves, the way they're shaped. It will just run up the, got it the wrong angle here. It just easily wets itself very easily all the way through that whole strand, the way the leaves are shaped. It's very clever. One of nature's clever little shapes. Reverse it, go the other way. God, it's so hard to do this in reverse. Everything's flipped. I should have flipped the camera software, but I didn't. What a pain in the butt. There we go. See it coursing through the stuff there. Very interesting. But at this magnification, you're not going to see a whole lot in this stuff, except maybe a small insect or some other arthropod or something, but it's always nice to look at anyway. It's cool. Cool structures. Let's go way in here and see if we can find anything. I think you're going to need more light here. Let's use another accessory light. There we go. Zoom in down there a bit. The tray I'm using is blocking a lot of the light from below. It's not my typical petri dish thing. I need to clean them, so yeah, little leaves. So I'm going to toss this in some water. And you have any little tardigrades that are possibly dried out or dormant. See if they'll rehydrate in a little bit here. And then I've got oak. I had some oak leaves and stuff last time. Coast live oak. And here's a slightly different sample of it, but it's really cool. You can see the groupings. So that you see this kind of waxy looking. This is the underneath part of the cell. It's a little less protected than the top part of the cell, but it has this kind of a dull, waxy look to it, and it helps. The plant lives in full sun, chaparral, oak woodland, so it's constantly getting bombarded by sunlight all day and heat. So plants have all these strategies to. Oh, see you, Robert. Sorry I missed you leaving there. Thanks for coming by. Some of these actually have fuzz at the little axils, the little junctions of the leaf venation. That's actually kind of fun there. You can see the venation is actually depressed. It's not sticking out. It's actually recessed. Very 3D looking. This one has the nasty spines on it, so animals don't go around browsing the leaves indiscriminately. So, tons of little pointy bits to discourage you. And I get stabbed by them all the time just picking these leaves. They're quite sharp. And leathery, waxy. And you flip it over to the harder part. And I adjust the focus here. So Similar appearance to the bottom, but they're even more kind of waxy. Not so shiny in this one, more dull, waxy for all the extra water protection the leaf needs to not dry out. Now they still have, I'd have to go way close to find them, but they still have the stomata underneath, or stoma singular, where it looks like these little, like a little mouth shape, where the cells allow air to come and go, and also water vapor. There's, as you know, part of the water cycle is plants transpiring. So we respire, plants transpire. They respire as well, but the transpiration is the is giving up of the water to the environment. So going up through the roots, through the plants, up through the leaves, and out. And at mass quantities, it's a huge part of the water cycle. Is plants transpiring, water evaporating, coming out of the leaves and going up into the local environment, and then the further up, if it's, you know, the temperature and conditions are right, go up into the upper atmosphere. But it's a very important part of the water cycle. But plants also go through, you know, they have photosynthesis under light, but at the same time they also are doing some cellular respiration that we do. But when they're in full-on photosynthesis mode, they're doing a hell of a lot more photosynthesis than they are respiration. And cellular respiration is basically, there's a simple equation you can draw. It's just basically, like for photosynthesis, it's water plus oxygen and chlorophyll and energy and all that kind of stuff gives you water, and CO2 gives you carbohydrates and oxygen. So that's why plants are good for your local environment in your house or your office during the day when there's plenty of light, because as they're doing photosynthesis, they're actually giving off a lot of oxygen to the room, as well as giving you like a nice smell from the plant itself sometimes. So while they're making sugars, they're also giving off oxygen. And cellular respiration is the opposite, basically. You're consuming sugars and oxygen and giving off CO2 and water. And that's what we do all the time. Because we're not autotrophs. We're not making our own food. We're heterotrophs. So we're basically consuming, whereas plants do both, which is why they're so wonderful. So that is the leaf of the live oak. But I also got one of the acorn caps. So I can get a better view of it here. It's kind of shaggy, scaly acorn caps off the acorn is already dropped off. And you can see the scales there. This is the part where I broke it off the branch. I can grab this just right. I can turn it a little bit. You can see it better. There's the scaly acorn cap. So, like a little hat, the acorn grows from inside here, and scales will eventually fall. The caps will eventually fall off the tree, but they're pretty durable. The important thing was the acorn itself got to be distributed, so the plant, you can grow a new plant. But there's a little hat for the acorn. So, and there are different amounts of scaliness and color in some of these different species of oak. So that's fun. I did get some fungi yesterday. This one was quite colorful, but has already kind of dried out a little bit. It was forming some type of kind of a foliose shelf fungus. It kind of spreads out in a circular pattern instead of a typical shelf pattern like the turkey tail fungus does. And so this one was on the edge of the log, and you can see the turkey tail banding there. but this one tended to grow more flat on the log and not stacked in shelves, like a typical shelf fungus. So they have some have slightly different growing patterns. Get this. Turn that down a little bit. Maybe kill that. That's my backlighting. See, there we go. It's more color. Backlight kills the color sometimes. So that's the top part, the colorful bit, and then you can turn it over and see a little more of where it was attached to the log. A little bit of moss there that was stuck on it. And I'd have to zoom in quite a bit to see the hyphae, little threads that attached it. But I had no luck yesterday with mushrooms. It's kind of, we had a lot of rain and cold weather and stuff, but I think the fungi this year were kind of confused. I didn't hear much about the Santa Cruz Fungus Fair because it was a virtual one again. They didn't have an in-person one. But I didn't hear much about the what was going on with the quantities this year and how successful everything was, and if there was a bumper crop of mushrooms everywhere, or if it was more just kind of a slow year. It's all about the timing and the water and the temperature and light and stuff. But yeah, so that's that one. I don't know the species of this one, but I have a picture of it I can show later if I can find it. Very colorful. This was more of a typical shelf fungus, and you can see the shelving as it's growing on top of itself in layers. And you can see at the bottom has the gills, like a lot of mushrooms do, not all of them. Some have pores, some have gills, some have false gills, like chanterelles. But yeah, kind of typical white, chalky, fuzzy fruiting body here. So fungus. And if I had planned more, I would actually already have spore prints on different color cards so you can see what the spores look like. And you get a spore pattern, which is kind of interesting. It's kind of artistic. But there's the shelf fungus. Now if I can zoom in. It's funny too, because on the way to collect some of these samples, there were a bunch of wild turkeys out. And I got some pictures of the wild turkey's tails, which look just like this. So turkey tail is a great name. It really does look like it. Zoom in a tad here. Let's go to one of the more colorful areas. You can see the banding, concentric rings, and the banding. It's about as much light as I can get on this specimen here. But you can see when you get really, really close, the pattern breaks up. It's not just these solid lines. You actually see the individual threads of hyphae, which is basically the fungal equivalent of the bulk of the fungus is underground or in a log or sometimes aerial if it's wet enough. And fungal hyphae collection in thousands and thousands of threads collecting together to form the body of the fungus, just like the plant has, roots and a stem and branches and leaves and stuff. The fungus has hyphae, and as a collection of hyphae forms a thing called a mycelium. So a mycelium is a single organism, collection of thousands or sometimes millions of hyphae. And there are huge ones that are like acres in size or bigger, where it's one giant mycelium that has grown for hundreds and probably thousands of years in some cases. That's the same organism. One giant mycelium could take up underground a whole huge landmass, and you'd never know it's there because it's mostly underground until it has a fruiting body of some kind, or you dig in and you see the mycelium underground. So if you watch the show The Last of Us, based on the video game, with the cordyceps fungus attacking people because of global warming and mutations and stuff like that, when they cut into the skin of an infected person, you see all these fungal hyphae. The problem is the show and the game try to make it more interesting and less scientific because it's a show. They got to get you going. Fungi don't move. They grow to go place to place. The only fungi that really move are the slime molds, where the individual amoeboid cells can rally together at some point with a chemical trigger, and they have these great time-lapse footage of these kind of fuzzy yellow ones or gelatinous yellow ones. They're kind of like one's called witch's butter, grows around here, and they get they can be individually growing, and then they'll get a cell signal of some kind, some chemical signal, like sometimes cyclic AMP, which is a form of ATP energy, is sometimes going to be a trigger. And again, I read about this years ago, so I forgot the details. But they get a rally call to assemble. So kind of like a Voltron or some kind of cartoon where all the robots come together to make a massive robot. The individual amoeboid cells come together and not only group together to form a big structure, but they also specialize and modify themselves to be firm, hard structures. And some are more for reproductive purposes, some are for structural purposes to form a basic fruiting body like a mushroom. But it started out as this gelatinous or liquidy looking amalgam of cells because they're individuals coming together. It's really fascinating stuff. Anyway, so they don't move except for those guys. Mycelium it spreads by growth. So in the case of the Last of Us, it's invading your body. It's growing just like bacteria will spread through your body through constant cell division to spread. The hyphae will continue to grow more and more threads in different directions, just like a tree root grows, pretty much. The rootlets and root hairs and stuff. But that's its movement. It doesn't actually have any way to. If you watch all these little microorganisms I have under the slide here, they have flagella, they have cilia, they have all kinds of little tricks to move around. Hyphae can't do that, but in the show they show these tendrils coming out of the mouth and waving around and stuff like that. That's just, that's purely entertainment for the show and for the game. I guess I haven't played the game. Hyphae do not move. They could be hanging and blow in the wind. That would be some kind of movement, or water would make them move, but they do not move on their own, like tentacles or tendrils of a small, underwater creature or whatever. They just grow. They move by growing and forming a massive structure and branching out and spreading and stuff. So they're pretty efficient at that, but they're also not super fast. In the show, they have time to infection for like a bite, a human infected bite with the fungus to the head area or neck is like within minutes. You're basically compromised. And then torso, so many hours, and then on the foot, within a day, you're pretty much compromised. And that's assuming they're talking about the growth of the organism. Well, it would take weeks if your body's immune system was jacked up to even get anywhere near your head from your foot. To grow that much mycelium from that thing would take a hell of a long time. So, but they're also, I think, possibly talking about the drug effects of the cordyceps, because like you can get in ants, the true story about ants and cordyceps, where they produce chemicals and grow in the ant's brain to make the ant behave a certain way. And one of the behaviors is to make the ant, doesn't kill the ant because it needs to reproduce. So it makes the ant go to a higher altitude than it's used to. So it goes way up on a plant. So when it finally dies from the cordyceps and a fruiting body sprouts out of the ant's head, it's up in the wind area. So the spores will be carried by the wind because the fungus made the ant go to that part of the plant where it had a lot of exposure to air, to wind. It's a very clever adaptation, not for the ant, but for the fungus. And in this one, it's supposed to be they're kind of mixing up cordyceps and ergot, because ergot's basically like a fungal LSD, which is supposedly the whole backstory of witches taking ergot, and it's a poison, but on smaller doses it's a hallucinogen like LSD. And they were also experimenting with other things too. So they're thinking that this fungus in the show is actually also using chemical stuff to kind of make you crazy and violent and disoriented, and which is why they run around rampant like the infected people in 28 Days Later. It's not really a zombie movie; it's just infected people. But anyway, it's an interesting show. It's a lot of fun, but the science part of it is, as usual, it's entertainment, so you can't take it everything with not even a grain of salt, more of a molecule of salt. So, but it's fun. It's just good to know that it's not an accurate depiction of how things work. But unfortunately, some people get their science from social media and bad news sources and TV shows and video games, and that's a very bad idea to do that. I make plenty of mistakes on these channels, on these streams. I do my best not to make too many, horrible errors and give wrong information, because some of the stuff's from memory and some of the stuff from stuff I read recently and just general biology principles and chemistry and stuff. But everyone makes mistakes, but there's a difference between making mistakes and not caring about the facts and twisting the facts to suit your agenda and teaching science that's not really science. It's more of getting people to do what you want or believe what you want and not the actual facts, because science and the crazy natural world, as you've seen some stuff I've shown and plenty of other things online and nature shows and TV and stuff, it's crazy enough. You don't need to embellish it that much. Now, Last of Us has to make an action show out of it, so you have to make stuff go faster. It makes sense. Just like 28 Days Later, when you got infected, like 20 seconds from contact with the infection, you're pretty much compromised. And it's like, well, nothing works that fast, except for some chemicals do, but not a biological organism of any kind. It's not that fast. Even the toxins of a bad snake bite tend to take at least a few minutes that kill you before you die, or die over a couple days, depending on what you got and how much. But movies and TV shows and video games have to make it exciting, so you'll continue to watch and be entertained. So as long as people know that, it's fine, because I look into it. I think it's entertaining. It's fun. You just don't want to get all your science from pop culture and from non-credible sources. So that's why I always say, if you guys have questions and watching this on the playback or live stream or whatever, feel free to ask questions, or if you thought I did something weird or I said something odd, or you have corrections, I'm always up for that information, because no one's perfect and science changes all the time. That's why a lot of people don't like it. They say, you guys keep changing your mind, can't you be consistent? It's like, well, no, the whole point is you go where the data takes you. Whereas something like religion is stone tablets, essentially. Also metaphorically, it doesn't change for thousands of years. So you have to change with the data. We would not have the platforms we have to talk about science and technology and medicine and all kind of stuff without the rigors of the scientific method and peer review and verification and you know, point out the liars and the cheaters and stuff, because if things aren't precise, things don't work correctly. We don't have satellites that work. We don't have the internet. We don't have computers that are consistently working, although they do break a lot. That's mostly human error from programming and maybe faulty tolerances on silicon chips and stuff. But in general, the amazing stuff we're doing because of the massive leaps in science, even over the last 20, 30, 40 years, is because of this information, the way it's presented, and the fact that it matters to be correct, it matters to be accurate, it matters to go with the data and not cheat to get your stuff published and to make more money. And you know, it's all about the pursuit of knowledge and the benefit of that for everybody after that, not to get an agenda across, and not to, you know, all the other bullshit that goes along with that stuff. So it's a human nature thing to do that, but it's also very bad. And scientists who do that are pretty much useless to most people. I mean, there are plenty of junk scientists out there that people love watching their channels and go to their seminars and buy their books and look at their appearances on TV shows and stuff. And it's, you know, Carl Sagan was a great guy, and he was very legit up to his death from cancer. You know, one of the heroes of not just astronomy and general science, but even concepts of space travel and exobiology and all kinds of stuff. So interesting, really interesting guy. Got to see him talk once in person, which was pretty special. So yeah, good stuff. Okay, so that is the shelf fungi, which are always fun. Got a chance to show that. I did get some other, I may have shown this before, but this is maidenhair fern. Very, very delicate. I mean, it looks like a little black wire. It's so thin and kind of plasticky looking, but these are the stems of the maidenhair fern. And if you look at the leaf shape, this one's a little flattened out. It's starting to curl because it's starting to dry up a bit. But if you know trees at all, this looks quite familiar to some people. There's a tree called the maidenhair tree, also known as ginkgo biloba. So they're not related at all, but it's just funny because this is that same shape of that leaf. It attaches a little bit differently, but it has that kind of fan out leaf shape of the maidenhair fern. And is also a very interesting tree for lots of reasons, not just the medical use of it, but the way the leaf goes with the veins coming from a center point and radiating out in a fan pattern. It's just like a ginkgo leaf, just on a really small scale. And so really interesting. But of course, this is the fern, so this is a non-flowering plant that produces basically, it reproduces through basically like a spore type thing. I've never seen the reproduction of these guys, but I used to read about this stuff. There's all kinds of diagrams and books if you look them up. Ferns are really interesting. But this one's very, very delicate. If I can zoom in on that venation a bit, you can see closer what I'm talking about. Let me fix that contrast a bit here. See the venation there. So if you know ginkgo leaves, it's like a micro ginkgo leaf. It's very interesting. So maidenhair fern, maidenhair tree. Not related, but just similar appearance and structures on a purely visual level. Let's go a little closer and see what we got here. See the veins quite well there, and the kind of texture of the leaf. So these are, the long ago, basically prehistoric plants They're still vascular plants because they have veins and structures for nutrients to go through the cells. There are plenty of non-vascular plants that are very primitive out there. But these are the more primitive vascular plants, and they also tend, because they're delicate, they tend to be near water or needing moisture, at least humidity and stuff. So they're in various forests, redwood forests around here, and shaded oak woodland areas with lots of water. Let me give you another fern here I collected. This is a piece of a wood fern. This is your typical fern look that everyone's used to in the world. It's got this thing, and there's even a fractal that pretty much does this fern perfectly with math. It's very interesting. I forgot the formula for it or what it's even called. I call it the fern fractal, but they used to have shirts for it when fractals are all the rage. And there's an actual calculation to build this in math, all the way from the growth pattern, from these, going down to the fine point, to the triangle shape of the overall body of the stem, to all the little branches. So fractal stuff, pretty cool. And when you get closer, of course, there's a lot more detail and interesting details, structures, and venation in the leaves. I don't remember ever seeing these sporulate either. So I did have some polypody fern last time from a different hike that had the little spore structures underneath, but this one doesn't have that. So do I still have that piece of fern? I wonder. I did have a bag in the fridge I pulled out that had some other older samples in it. Yeah, there it is. Here's the polypody fern. So for comparison, and because it's sealed, it's still nice and green and damp. That's the wood fern. This is the California polypody fern. And if you look from the bottom, from the top, you can see, move this around a little bit. See, it's all lumpy from the top point of view. There's stuff at the bottom bulging out from the leaves at the top. Like, oh, that's interesting. What's going on there? So when you flip it over, also you can see the way the growth pattern is very different on this one. It's got this kind of like fingers. That's the polypody name, is because of the way the fingers branch. They're kind of this alternating branching pattern. See, they're not lined up, they're alternating, and they're kind of finger-like, lobe-like little leaflets. So you flip it over, look at those spore packets. And they actually look like little green, almost like insect eggs. If you look at caterpillar eggs and stuff, they have this similar kind of appearance to them. Really interesting little guys. And some have protective covers, like you have your beach umbrella or your pool umbrella. Like, say you have your table you sit at the pool, and it has the little pole that comes up through the table, and then it has the umbrella that goes around you. Well, imagine these clusters of little spores gathered around there's a little center point that comes up, and a little umbrella structure that goes around it and forms a nice little sheath. So rain and animals rushing by and brushing on the plant don't prematurely spread the spores when they're not ready or too much at a time. It wants to be efficient in how it doles out the spores. I would imagine. I mean, it's not trying to give attributes to a plant, but but basically they do have protection mechanisms, some of them. This is just a naked one. It's just a little kind of sticky cluster that can break off and spread. But in some they actually, and I think it's the sword fern that has that. I haven't collected any sword ferns in a while. So I'll have to collect some of those. There's also a chain fern, a couple local ferns. There's a This maidenhair fern is 5-finger fern, which looks like maidenhair but has this big kind of a hand pattern to it. Goldback fern, which is this tiny looking, it's like almost like a wood fern but very tiny, cute little thing. Wood fern, chain fern, polypody fern, and a few other ones. So yeah, it's not just mushrooms that produce these little like spore-like bodies. The ferns do it as well, and of course viruses do as well. They also form kind of a little spore structure, micro spore structure. So that's the ferns. I've had enough time for this moss soaking. I bet I got a nice tardigrade in here. Should have at least one. I'm going to do a quick couple siphonings here of the water from the fern. Let's see if I can get a tardigrade in here. Tardigrades are also known as moss piglets by a lot of some German biologists and stuff, because they tend to grow on damp moss for their to look for their food. But as you know, moss dries up easily on a regular basis, so tardigrades actually have this amazing mechanism to be able to dry out to the point of being like in a pure kind of stasis, not just like a torpor or hibernation like a hummingbird would do or a bear. They're actually, for most purposes, essentially shut down biologically. And they've even taken them up into space and exposed them to a vacuum, and they survived, and it's just crazy little guys. So I'm gonna leave the fern here for a bit longer while I look for the tardigrade, because I'm gonna go pretty fast. I don't want to make people dizzy. Do a quick tardigrade scan. See anything yet, but they're very big on a slide like this, so they should be easy to spot, which means I can run a couple different slides until I find one. Nope. Nothing there. Okay, I think I'm going to have to make another slide. And you don't always find them. You're not guaranteed to find these guys, but they are pretty much every single time I've collected moss, whether it was dry or wet, and I soaked it and I gave it a few, anywhere from a few minutes to several hours, I was able to find a tardigrade. In fact, I think this is the last time I had one on stream, a couple streams ago. You could actually see the tardigrade starting to revive as I found it on the slide. It's very, very sluggish, almost not moving at all, because you do occasionally find dead ones. But this one was just starting to come out of it. All of a sudden became super crazy active. It was all over the place. But it was fun to see it wake up. You know, it went from totally out of it to pretty wild, moving around. Let me change the ambient stuff here. This track is getting a little bit old. Let's try a different one. Let's see what happens on this guy. This one's called Change. Last one was called Eons. Okay, so no tardigrade on that slide. Let's make another slide. Do a quick look on this one. So you're not seeing it on the stream because I'm just going so fast. I don't want to make people dizzy. Not there either. Okay, so far it's a bust out, but I'm not giving up that easily. These guys are always worth finding because they're so cool. So I'm gonna try. Let's see, where to put my. I do have one remaining petri dish over here somewhere. If I can put a bunch of stuff in the dish and put it on the dissecting scope, you actually see tardigrades at a much lower power, but it's fun to watch them in their natural color at low power. Kind of transparent. Kind of transparent, kind of opaque white, semi-transparent. So let's transfer some of this debris to a petri dish. Oh, come on, you can do that. Some water. Move around a little bit. More water than that. And let's put it under here. And just see what we can see here. Sometimes you find insect larvae in here, tardigrades, all kinds of things. There's the nice shot of the moss underwater, all spread out. Get a little bit of a glare from the light reflecting in the water, but it's okay. So tardigrades can be, is that one right there? They can be about just under a millimeter in size, so they're pretty big when it comes to microscopic stuff. But they're small enough that you still have to hunt for them, especially on a scope like this, unless you go to real high power, and then it's really hard with all the water sloshing around. But sometimes they're still clinging to the moss, and sometimes they're just kind of floating around, swimming in the water. Let's do a little backlighting, that might help a little bit. So you guys can see it better on the camera. The camera is less forgiving than the microscope optics are. Backlight kind of messes that shot up a little bit. Basically looking for a little wormy guy with 8 legs. Tardigrades have 8 legs. 2 of them on the back segment of the body, which is funny. Just 2 legs on the very end of the body. And what's fun about them too is, as I've said on other streams, you know, with a whole bunch of animals. Oh, wait a minute. I don't think that's one, but God, it's really shaped like one. Let me just investigate here real quick. Yeah, it's just debris. But they're so specialized. They're in their own phylum. So if you understand the Well, they used to call it the Five Kingdom system. Now it's been changed a little bit. The top end of the table of organisms is basically, it's called domain. But essentially, the older Five Kingdom model was, kingdom, phylum, class, order, family, genus, species is the order, and there's the mnemonics you can use to memorize the order of it. So humans, Homo sapiens, are in the old model, kingdom, Animalia, phylum, Chordata, subphylum, Vertebrata, because chordates means you have a dorsal hollow nerve cord. You have Vertebrata means you have a vertebral column to protect the nerve cord. Class is Mammalia, so all mammals. Order is Primates. This looks like primates, but it's pronounced Primates. And other people don't want to hear that. They say, "Oh, we didn't come from monkeys." It's like, it doesn't work that way. We're all primates. Get over it. Primate doesn't mean monkey. It's classification of certain type of the great apes, the monkeys, the various other things are all in the primates. So we're a primate. And then so that's order. And then family, hominidae, so hominids. And then genus Homo, and then species Sapiens. Now there's subspecies in a lot of things. So technically, I don't know, maybe this changed recently too. I don't follow anthropology that much, but it used to be Homo sapiens sapiens, because there was a Homo sapiens something else, subspecies, and I forgot if they got rid of that, if they still keep that. But that's how it works, basically. The new one is I believe kingdom was replaced by domain, and similar breakup of the top level stuff is basically fungi, protists, which covers kind of all of the single cell plants and animals. There was one called Plantae, which was Plantae. That was the plants. Complex algae, plants, ferns, all that kind of stuff. And then you had Animalia, which is all the animals. They're not single-celled animals for the most part. They weren't under the protists. And then you had what used to be called Monera, which was all of the bacteria and bacteria-like thingies. So if you go into details on the different types of cells, bacteria are called prokaryotes, meaning they don't have a nucleus. Their genetic material is in the cytoplasm of the cell. They usually form these little rings called plasmids. We actually did a experiment in college where we actually used restriction enzymes to snip up the plasmids. Use a shock mechanism to for the bacteria to accept kind of basically their pores, for lack of a better term. Their pores open up on the side of the on the cell, and they're able to immerse the new gene fragments into the matrix, and they're absorbed by the various bacteria, and then with a different mixture of restriction enzymes, you can have them add new genes to their plasmids. So you can actually add like penicillin resistance to bacteria in a very simple lab experiment. The enzymes are expensive because they're specially made and isolated, but you can do it in a basic biology class with a certain number of tools, and do genetic engineering with bacteria. It's very interesting. So anyway, that's how they're easy to tweak like that. You don't have to deal with a nucleus; you just deal with the cell itself. Eukaryotes, which means true. Karyo means like kernel, or It's Old Latin for a kernel, and kernel meaning like the nucleus. So in eukaryotes, compared to prokaryotes, we're all eukaryotes, animals, plants, all that kind of stuff. We have nuclei. Our genetic material, our DNA is in the nuclei. The RNA is in the body of the cell, used for processing, protein synthesis and all kinds of stuff like that. But that's what separates us. So they're the very, one of the main classifications used to be put all the prokaryotes into one group. That includes bacteria, blue-green algae, all that kind of stuff. And so that's basic taxonomical classification of organisms. I forgot where they threw the viruses, because technically viruses on their own, standalone viruses, isolated from any cells of any kind, they're not even technically alive. They're basically genetic material packaged, almost like a spore of a plant or a fungus. But even a spore or a plant of a plant or fungus has more going on than a there is some cell activity in a spore, even though it's dormant usually, compared to a virus, which is literally just RNA or DNA packaged in a protein coat and some other protection mechanisms, so it's a durable structure. But by itself, without a host to reproduce itself, it cannot do anything. So I forgot where they lumped those guys into the taxonomic stuff. It's been a long time since I studied viruses, but anyway, long story short, that's how you separate out all the organisms of the world: plants, animals, fungi, all that kind of stuff. And so tardigrades have their own phylum. We're chordata, they are tardigrada or tardigrada. And I think there are about 1,000 species, maybe 2,000 species, something like that. Which is crazy, because you go to something like arthropod, phylum Arthropoda. That's the crustaceans, the insects, the spiders, the scorpions, the blah 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 blah, all kinds of stuff in there. And it's a huge phylum. But tardigrada has basically just a handful, essentially. Yeah, I'm not finding any tardigrades in here, so time to get another sample of moss. And it could be that there's none in this one. I'm just going to go ahead and throw it into the same cluster of stuff. And a little more of this fluid that I shook off it, in case it's in the fluid. I'd expect to find at least a dead one, or maybe a fragment of one or something. Okay, this is a much bigger cluster of material. Not seeing any on the thing itself. Yeah, I'm looking for the moss piglet. Let's see if I can find it. This track on this one is really dull. Pick another track. There you go. Fractal. This one's interesting. Very spacey. Okay. A lot more debris in here now since I moved some more water and moss over here. Scan the debris field. I mean, this is, in a way, this is similar to what they do in your medical labs when they take a blood sample. They're looking for certain types of white blood cells or certain types of parasites. They're scanning a sample that's obviously not as liquidy as this, or a pain in the ass to actually focus on. But they're doing a similar approach of scanning through an area, quickly and using their visual memory to things that pop out and go, oh, that's that. You got, 20 eosinophils, 20 neutrophils, 3 basophils, or whatever they happen to be looking at. Let me have some debris on this thing. And in this case, we're looking for little wormy looking tardigrade guys. To give you an idea what the size I'm looking for, let me zoom out a little bit. Something so this clump of debris right here, I'll put the cursor down here. Tardigrade would be about that size, maybe a little bit bigger in this debris field. So there's a lot of stuff here you can just immediately reject on appearance, just because it's not big enough or the shape is wrong. But when you see them, they're funny little cute things at this scale. They're not so cute if you had to face one at life size, but they're pretty aggressive little buggers, but just constantly stabbing things and eating things. So I'm not seeing any here, not in the loose liquid or clinging to the moss. Sometimes you get other cool, like, larvae and stuff. That's just debris there. More debris. Kind of hard to show up on this camera. Let me turn the backlight on again. It's a pretty thick cluster of moss right here. And last time I did this, I found like this little earwig larva. This little insect larva was in here as well, with little jaws. It's on one of my videos there. Because animals pass through here and will shed exoskeletons and stuff. Sometimes you get weird pieces of insect or spider body parts from sheds. Yeah, I'm not seeing any. No tardigrades. One other thing I can try. I did shake this quite a bit in the cup that I used to rehydrate this. It could be in the water I have left over. So I'm basically going to do a quick pass through the edge of this thing, and I'm going to go back to that water sample. What was that? Some kind of piece of something. All kinds of weird shapes at the scale. Okay, no tardigrades there. So let's do this. I'm going to dump this water into here. So the petri dish is mostly empty. Dump the rest of the water from the cup. Do a quick scan of this guy. Because you never know. Might be in here. See if I can focus it better so you guys can see it. I might have to zoom out too because it's awfully close. Okay. Let's do my little search pattern. Some debris, useless debris, nothing in the middle, you can see. So like this guy would be at the right size, but it's not one. But this is the kind of the size that they are. Lots of look-alike junk. Yeah. There's one more little bit of water, just to be thorough. And we'll try this last bit, because why not? What is that? These forceps are so huge. I gotta find my micro forceps. Something odd there. Didn't even find a dead one. So even times I thought I struck out, I at least found a dead one. This goes to show they're not just all over the place. You do have to pick a choice piece of moss. I don't even find any insect larvae or anything. Like one time I found some mites in here. One was even a tick. You know, the tick's like a large mite. But I can't even find anything like that. It's funny too when you get some protozoans in here as well. See them swimming around all different directions. Oh, wait a minute. What is that? Oh, no, it's flat. That was a flat. It's like when you go bird watching, you get used to certain bird shapes, and then you look at a tree and a leaf. A cluster of leaves has the exact shape of a bird shape you're familiar with, and you go, hey, that's not the bird. It's just a leaf or a series of leaves. It gets very frustrating sometimes trying to find things. I usually find at least some other little living things in here swimming around, or pieces of things. All I've got is bits of moss, and that's about it. I mean, some of this debris could be tardigrade dung. I've seen videos of them making quite a mess. Not a thing. Not seeing anything swim around on its own either, just moving when I move it. Well, it was worth checking. What do I have left in this little bag of stuff? Hey, Sensin, how's it going? Got some, I didn't make this music. This is an ambient generator because I can't be bothered to make hours and hours of ambient music. It's too... Although I should use your Holon app, the Holonist app, to do this. That would be kind of a good idea. I didn't even think about that for the live stream. And I can give you a plug on here too, so people go look at your app. I haven't had a chance to use it since you gave me that code a while back. But it sounds like a great use for live stream backgrounds. Just let me see if I have any extra moss in here still from before. Nah. Some mushroom bits. What's in this tube? This is so small. I have to put it on here to look at it. I think this is some lichen bits. Yeah, this is cool. I didn't show this last time. This is nice bits of kind of the old man's beard lichen that hangs off of trees and stuff. And you can see how delicate. Let me get this light fixed here. It's a little too intense. And this net, it does this net formation. It's very similar to some of the growth of arteries and veins and stuff, and even capillaries around your kidneys and other organs. Get this kind of a net-like structure. It's very delicate. And again, this is a lichen, so it's the body of a fungus with the color coming from the algae living inside of it. So it's a what you call a symbiotic relationship, and in this case it's a favorable one. So the home provided by the fungus and the food provided by the algae doing photosynthesis inside, giving it the color, and they work very well together. You know, there are plenty of other symbiosis relationships that are bad, like parasitism, parasites. That's one version of it. The other ones, like one's called commensalism, where basically one animal benefits the other animal or plant or whatever doesn't. but it's still a relationship because the one has to be there for the other one to do its thing. And there's several other versions of that. I forgot some of the terms, but look at that little net structure. That's really cool right there. Zoom in on that. Pretty cool. So that's just a little lichen sample from last time. I think I forgot to show it. And then I've got this guy. Let's see if I can get a close-up on this guy. This is it's called bed straw, even though it doesn't look like straw. It's not a grass. It's a type of that. Its scientific name is Gallium aparine. See those little spikes there? Those little white spiky hairs that are all along the. They're actually hooked in this case. You can see there's a little down curve on them. If I can get the contrast on this better. Maybe this will help. Yeah. Let's see. Maybe this will help here. I have to zoom in on them to show it better. There you go. So those little hooks there look like rose thorns, microscopic rose thorns. So the whole plant is covered with these. And it's just a very soft plant that grows in the areas kind of like where I found that newt. And some of the mosses and ferns. And it just looks like kind of a boring little herbaceous plant sticking up. But when you touch it and pull on it, these little things grab you. Now your skin is pretty tough, so you don't feel it, like it's not a painful thing. But those little hooks are really good at grabbing your hand, animal fur, anything. And you know, acts as a way of distributing the plant. Even though it destroys the plant in the process, it can actually pull the plant and make you drop it off somewhere else with any seeds or whatever it has, if it's fruiting at the time. You know, you get this with like burr clover, which is an extreme example. In that case, it's actually the seed pod, or basically a fruit in this case, carrying the seeds, is covered with all these little thorny bits. And you know, and another one called there's another one called goosegrass, which is similar to this, but a French inventor who invented Velcro got his idea of hook and loop fasteners from goosegrass, essentially. And I think it might be the same genus as this one, but you can see the hooks, and then anything becomes a loop, like your hair or fur of an animal, or you know, any kind of thing it can get its little hooks into for dispersal. So it's very clever. And then Velcro is like the universal man-made fancy version of that. But yeah, so that's bed straw. Very interesting little guy. And what else do I have in this little tube that I forgot to explore recently? Stuff's really crammed in here. I think it's just more stuff I already showed. It's kind of in a clump. It's hard to get it out of here. Ah, come on. Yeah, I showed this. It's just more maidenhair fern. Another clump of maidenhair fern. This is still kind of wet. And another fern, more of the wood fern. I had some goldback fern with the little yellow spores on it, but I think I misplaced that. Where I put that thing. It's another type of lichen. This is a what you call foliose lichen, leaf-like, not net-like or strand-like lichen. Foliose. This is like you can have foliose fungi grow like this, because this is a fungus body. So here you can see the spores collect on the outside in little clusters, kind of like on the moss. I'm sorry, on the fern. And then these little spore areas can blow away or be eroded away with some touch. Some of them, you get different fungal groups forming lichen associations. So some of them actually form these little spore cups, like you get in some of the different like the Ascomycetes, the cup fungi and stuff like that. They can form these little cup structures, and then the spores will form these little cups, micro cups on the surface of the lichen. But in this case, they're just like these little kind of grainy blobs, not really in a kind of cup thing, more like in a cluster, like again like the cluster of stuff on the bottom of the fern I showed, the polypody fern. I think that's all the samples I had. These are all the ones I had handy. So I got to use some I forgot to show last time. That was cool. Oh wait, and I have another tube here. Let's see. What's this? What is this one? Ah. This is a piece of western hemlock. Poisonous plant. When it's young like this, it doesn't have the kind of purple splotches at the base. Diagnostic of the hemlock. But it's the kind of thing that killed Socrates. Poison hemlock. Nothing really much to look at on its own. It's kind of boring looking, but it's also a water hemlock that grows in the ponds and streams around here. It's really nasty. These are some really interesting fuzzy, essentially catkins, I think is the best term for this type of flower head. Growing on, God, I forgot I picked this from. I think it's a buckeye? Might have been a buckeye tree. This one, I did so many weird collections yesterday when it was raining and stuff. I didn't make notes of pictures of everything to keep my memory straight, but you can actually see the pollen forming in this one. You can see the anthers inside there. But see how fuzzy it is. It's like a little micro version of a Grevillea or Protea that you get down in New Zealand and Australia, with like this kind of feathery, furry kind of flower head structure. But yeah, you can see the forming anthers down there, nice and yellow for pollen. That's pretty cool. So this would be a compound flower or inflorescence. Each little floret here is a flower. So each little floret has its little collection of anthers. It depends on the flower too. Some flowers have both sexes in the same flower, which are called oh, hold on a second. Yeah, I don't see any pistil in here with the stigma on it, which is the female part, where the little ovary is at the bottom. I'm just seeing male parts. So this this might actually be. Let me see if I can break up another one here. This might actually be from a silk tassel bush. There are a bunch of those growing in the hills around here too. I was just kind of randomly grabbing interesting little clusters. Silk tassel bush is interesting because it actually has separate sexes on separate plants. It's called dioecious. There's monoecious, which is like both parts are in the same flower. Dioecious, where you actually have separate plants that actually have the different male and female parts. So you need a plant of each to actually reproduce through sexual reproduction. It's a very, very interesting approach to that. Let me zoom in on those stamens. That's kind of cool. So the tips of the stamens are the anthers where the pollen is formed. They always have that kind of like a little coffee bean shape to them with a crease in there. And they don't look grainy yet, so they're probably maybe not producing pollen just yet. They're getting ready to, it looks like. They would have kind of a grainy look to them if there's pollen available. But look at that fuzz, that shiny, silky fuzz. That's why I'm thinking this might be from a silk tassel bush, because they have a characteristic look like this, but I never looked at them under a microscope. Pretty cool. Ooh, nice. Myrac patch. Thanks. Yeah, that'd be cool, man. Thanks. Yeah, this is a fun little free website called flowful.app. So flowful with one L .app website. And it's just streaming. You can sign up and do all this crazy stuff, but I always do the free version. And I'm just jumping through the different presets. This one's called Fractal, which is pretty cool and kind of dark. The one I use a lot is called Eons. The one I had before that was pretty boring. Let's see if I can find another one here. This one's called Mirror. But they're nice because they're all synthetic. You know, some of these have like piano bits and other bits in them, and you know, I like the synthetic stuff for this mood, for the exploring the micro world, or for astronomy stuff or whatever. Yeah, Flowful app is cool. There are a bunch of other ones like Black Hole and a few other ones that I've used. Free streaming ambient generators, but this is my favorite. Oh, thanks for dropping by, man. Catch you later. I don't know if you sent that out recently or if you left it a while ago, but sorry if I missed it. I was too enthralled by my flower explorations in the scope. Very nice, though. So I think with this kind of sad ambient music, it's time to bring this to a close. Oh, I did get a mushroom, actually. Hold on. Before I forget. My girlfriend had some weird yellow mushrooms for cooking. I've never seen one that was edible that looked like this. It's starting to dry out now because I picked it from her collection she bought yesterday, but look at that. That structure and that color. It's wild. Actually, my tint is off a bit. This stupid camera has been a little bit off all night. Fix that tint. It's supposed to be more of a yellow like that. That's more accurate there, I think. Anyway, let's close that because you get that stupid pop-up box. And a little more light. So it's just starting to dry. It's getting a little more wrinkly. But yeah, there's a lot of color here. And you can see the gills down below. This is kind of sulfur yellow, and then the stipe, the stalk thing at the bottom here called the stipe, is actually kind of creamy yellow. And you can see how fibrous it is. This is why, basically, I know I'm being really picky when I eat mushrooms sometimes, but I don't eat the stems on the mushrooms. It really bothers me when you go to a pizza place and they just cut the button mushrooms lengthwise without pulling the stem off. And it's always like a chewy bit, almost just like it's not cooked all the way or something. The cap is always the best part. When it cooks, it's very nice, and it starts out corky when it's raw, but then when you cook it, it gets a really nice texture. But the stipe never quite does it for me. It's quite annoying, so I always pull the stems off mushrooms when I eat them. But that's just me. But you can see how fibrous it is with all those little structures bound together. I forgot to look at what the kind this was, but it smelled kind of funky when she was cooking it. It's one of those funky mushrooms that, it's edible and it maybe tastes okay, but it smells kind of funky when you cook it. It's not a pleasant smell. So mushrooms are really good to eat, but some are just awful. It's just like some cheeses, like Limburger and stuff. I just don't get it. They don't taste good. They smell bad. What's the point? But yeah, interesting stuff. Yeah, some stems are more fibrous and nasty than others. I just usually avoid them if I can. But pizza places, they don't do that. You get button mushrooms all sliced up on your pizza, and the texture's wrong. It's just you bite into some bits. Unless they completely chop them up into like a chopped mushroom topping, then it cooks differently. But if it's just whole mushrooms sliced, where you have the whole curved profile of the cap with the attached stem, those are always extra chewy. I hate those. But yeah, cheese. You brought up cheese. I did some cheese stuff a couple shows back. Fungus on a blue cheese, and while I was looking at that, I also looked at some fungus I had grown on some bell peppers in the fridge I had to throw out. That was kind of fun. You can see the spore bodies, the sporangia. This one looked like little grenades. A cluster of spores formed like these little hand grenade shapes on the scope. It was pretty cool. But yeah, it's good stuff. I got boy, it was later than I thought it was. I did start to stream kind of late, but wanted to use these specimens up while I collected them. Got all wet and didn't want them to go bad, so it was fun. Got to say a few interesting things, but not as much as usual. So thanks for everyone for joining, and anyone who watches the replay. I really appreciate it. Feel free to leave questions, comments, whatever. And one of these days I'll try to have a more scheduled, structured show where it's just a time it's going to happen, instead of spontaneously, here's the show, check it out. But I don't know, sometimes I just feel like doing spontaneous stuff, especially when I have a new sample set. I can't really plan for when it's going to go. I just have to do it when they're fresh. But good stuff. So thanks, Lone, for showing up. Thanks everyone else who was here, Sensian and Manny, Dr. Synth, and Robert, Native VS. And who else was here? A few other folks were here. No Wagyu tonight. Wagyu's either busy at work or he's sleeping. But anyway, thanks again. I really appreciate it, everybody. And always remember to stay curious and enjoy the natural world out there, and don't let the bastards get you down. When you're having a bad day, go look at some plants or mushrooms or birds or whatever. Get some fresh air, go for a hike, and enjoy what you got. Lots of cool stuff around. So thanks again. I'll catch y'all later. See ya.